Hello everyone, my name is Omar Santizo and I'm part of Belmont's research group. Today, I will be presenting my research on improving undergraduate chemistry education, analyzing Redox analytes using a student-built Potentiostat. So an overview of my presentation today will be our purpose, the Potentiostat, improving education, biochemical and electrochemical education, implementing into the curriculum, build a portfolio, results, future work, conclusion, references, and acknowledgements. So our purpose, modifying existing equipment typically used by the maker community with a vision to bring a low-cost, accessible instrument in the undergraduate lab. Bring in biochemical perspectives that happen in the human body that relate with the reagent used in the experiment. So the low-cost instrument that I talked about in the previous slide is called the Potentiostat. The Potentiostat is a potentiostat that's able to read silicon. This Potentiostat is composed in under $30, and then something special about it is that it's pretty cheap and affordable for the school. If you compare original Potentiostats that are in the market, they run from $7,000 to $20,000, which makes it very difficult for schools like House of the Megaschools to buy one. So what is this Potentiostat composed of? So the TC stat is made of three components: the breadboard, the TI LMP 91000, and the TNC 3.2 control. The TI LMP 91000 is an amplifier circuit that is used to convert raw analog data into digital data. The TNC 3.2 control is a hub, a place where all the data is gathered that communicates with the LMP to give the user flexible data for analysis. And then the flexible data analysis will be it is converted into numbers, which in Excel is used to find calculations and so on. So how I mentioned before, this thermostat reads silicon. So what is a silicon? Silicon is a method used to understand electrochemistry or concentrations in a course called quantum analysis. So it reads three types of silicon. It reads reversible, irreversible, and quasi-reversible reactions. So how can the T-stat improve undergraduate education? The T-stat can improve undergraduate education by having the students create their own T-stat protocol, and this will introduce students to the field of electrical engineering. It also will tie with Professor Belmont's lecture on concentration. As analyte concentration increases, single detection by device increases. And it will also introduce the method that I have been saying, silicon. The experiment is also designed to correlate real-life biochemical applications for students to connect and understand experiment on a deeper level. And finally, the TNC will allow the students to understand electrochemistry, which is the movements of electrons. And then they will be able to take this concept into higher education, such as grad school or upper division courses. So my part of the research was to build a portfolio with different analytes, which was acerbic acid, acetaminophen, and N-acetylcysteine. So I will be talking about acerbic acid. So acerbic acid is also known as vitamin C. It's an antioxidant used to kill free radicals and treat scurvy. And this reagent or analytes reads irreversible redox reactions. So where do you find vitamin C? You can find it in fruits, vegetables, and so on. So where we're gonna correlate it to real life is by how vitamin C acts as an antioxidant, which oxidizes to give its electron to a free radical. So a free radical is a free radical is a bad component of health, which is found in fried foods, tobacco, alcohol, air pollution, and pesticides, which they could cause disease in a given organ or place. So the antioxidant will be giving his electron to the free radical, which will be neutralized, which then the immune system of our body will be able to kill this free radical and create it into waste. So now let's move on with acetaminophen. So acetaminophen is also known as Tylenol and is a pain. It helps for pain and fever relief. This will demonstrate a quasi-reversible redox reaction, and then also we have not yet found how to correlate it to real life. However, our vision is to correlate it in how acetaminophen helps with headaches. So that's our vision, and will be implemented in the future to come. So now we'll be talking about N-acetylcysteine. So N-acetylcysteine is another antioxidant. It prevents asthma attacks, can treat rashes and other skin problems. It can prevent liver damage caused by overdose of acetaminophen, and this will also illustrate quasi-reversible glucometry. So the way we're gonna relate this to a real life application is how N-acetylcysteine helps with the overdose of acetaminophen. So we'll be able to correlate these two different reagents and create an experiment where the students will be interested in reading it and learning about electrochemistry and any other analytical method. So results. So this is comparing our Potentiostat to an actual Potentiostat. So if you guys see here in this picture, you guys will see that there's a reversible, quasi-reversible, and irreversible cyclophotometry. And then if you compare it to our Potentiostat works. So this will be our reversible cyclophotometry, which we ran it with ferrocyanide. And then you guys will see this is a reversible, and it looks very similar to compared to the black reversible in this original graph. So now if you compare acerbic acid, which is vitamin C, you guys will see that the oxidation peak is over here. So then our if you guys compare acerbic acid with irreversible, it is very similar. So now let's compare acetaminophen. And acetaminophen, if you guys see on the left side, this is a quasi-irreversible reaction. And if you guys compare it with the blue one in this actual graph, it is very similar. So now, before I move on, I want to describe the calibration curve. So for each analyte, we made a calibration curve which tell us how reliable this Potentiostat is. And according to our calibration curves, all of the reagent is reliable because the R-squared value is higher than 0.98. And this is acetaminophen, this is N-acetylcysteine. And in this one, it's also reliable, is higher than 0.98. So that means that our Potentiostat is reliable for the students to conduct research. So implement to the curriculum. So how I described in the beginning, this will be implemented to curriculum. In 2019, Professor Belmont's research team had the honor to actually perform the experiment with ferrocyanide, and a group of students they were earning extra credit, and then they ran the experiment. So it was an experiment of 2 days lab period, and then they were able to create their standards solutions and so on, and they ran the experiment, and they got their calibration curve, and they were also able to get an unknown, which they were tested with precision and accuracy. However, however, they were graded with precision actually, but they were given extra credit due to being the first run. And then if we see the results, they were able to get the reversible reaction, and their R-squared value was a little bit lower than 9.8. However, we decided that it was okay due to being student run and our first trial with our experiment. So we decided it was okay, and it was going to get better as more students were able to conduct the research, not conduct the research, conduct the experiment. So future work is to create a written experiment for the students to conduct the research on their own, start having students conduct the experiment. Lastly, keep testing the Potentiostat with other analytes, for example, glucose. And then future works that will be continue, same thing as so target it and then so make a new experiment, real life biochemical and electrical application, which the students will also learn. And then conclusion, success. Belmont Research Group has developed the Potentiostat. It's low cost, and it will teach the students about electrochemistry, and they will be able to take it into graduate school and so on, learn about real life applications. And and then they will take this knowledge anywhere that they will, anywhere that they will conclude their career at. So acknowledgement, the device developed phase of this project was funded by the 2013 SCAP grant for undergraduate analytical chemistry research. The student experiment implementation phase is funded by IRA grants and a McNair Scholar Program and Office of Undergraduate Research at CSUDH. And a special thanks to Barbara Belmont, she's our mentor, Justin Hadaway, which is our other mentor, and Claire Ledon, which is my partner. And this is a highlight of our research. So we got first place oral presentation last year for student research, first place for poster presentation, UC Irvine, and third place our mentor Justin won as well for student research day in 2017, I believe. So these are references, and that's everything. So thank you, and hope you liked it.